Welcome to this presentation on experiments related to photosynthesis. We will explore different tests to understand various aspects of photosynthesis, from starch formation to the necessity of sunlight, carbon dioxide, water, and chlorophyll. This experiment involves 1. Boiling the leaf to kill it. 2. Decolonizing it in alcohol. 3. Softening it in boiling water. 4. And then testing for starch using iodine solution. The presence of starch is indicated by a blue-black color. In this experiment, we use an aquatic plant and observe the oxygen bubbles forming on the leaves under a strong light source. The steps are as follows. Place the aquatic plant in water and cover it with an inverted test tube filled with water. Place the setup under a strong light source. Observe the oxygen bubbles forming on the leaves and collecting at the top of the test tube. This demonstrates that oxygen is given off during photosynthesis. By covering part of a leaf with foil, we can show that sunlight is necessary for photosynthesis. The covered part of the leaf does not turn blue-black, indicating no starch formation. Using a bell jar and potassium hydroxide, we can show that carbon dioxide is necessary for photosynthesis. The steps for the test are as follows. Place the plant in a sealed bell jar with a dish of KOH solution to absorb CO2. Expose the setup to sunlight for several hours. Perform the starch test on one of the leaves. The leaf will not turn blue-black, indicating no starch formation due to the absence of CO2 results in no starch formation in the leaf. By using a variegated leaf, we can demonstrate that chlorophyll is necessary for photosynthesis. Only the green part of the leaf, which contain chlorophyll, will turn blue-black. The leaf on the left is a variegated leaf. The green parts contain chlorophyll and photosynthesis to make starch. The white part of the leaf does not contain chlorophyll, so does not photosynthesize. The leaf on the right shows a positive test for starch in the areas which contain chlorophyll, and a negative test for starch in the areas which lack chlorophyll. We can study the effect of light intensity on photosynthesis using the following steps. Place the aquatic plant in water and cover it with an inverted test tube filled with water. Place the setup at different distances from the light source, example 10 cm, 20 cm, 30 cm. Measure the rate of oxygen bubble production at each distance. The closer the plant is to the light source, the higher the rate of photosynthesis, indicated by more oxygen bubbles. Let's take a moment to test your understanding of what we've covered so far. Please choose the correct answers to the following questions. Also, please choose the correct answers to the following questions. In summary, these experiments illustrate key aspects of photosynthesis. The starch test reveals the presence of starch in leaves, while the oxygen release experiment shows that oxygen is given off during photosynthesis. Experiments on the necessity of sunlight, carbon dioxide, and chlorophyll demonstrate that these elements are crucial for photosynthesis. Additionally, the effect of light intensity on photosynthesis is highlighted by varying the distance of an aquatic plant from a light source and observing the rate of oxygen bubble production. Together, these experiments provide a comprehensive understanding of the photosynthesis process. Thanks for watching. To deepen your understanding of any topic, explore our comprehensive resources, including revision notes, past questions, gamified quizzes, and workbooks. These tools are designed to help you master the concept effectively and enjoyably.